Welcome to first lecture on Class 10 Science, Chapter 8, How Do Organisms Reproduce? Let's begin by understanding the definition of reproduction. So the production of new organisms of similar type is called reproduction. Significance of reproduction. So why is reproduction so important? Reproduction is very important for maintaining the existence of species in the ecosystem. Types of reproduction. So as we have studied in the previous classes, reproduction is of two types: the asexual mode of reproduction and sexual mode of reproduction. In the asexual mode of reproduction, the offsprings arise from a single parent without the fusion of gametes, while in sexual reproduction, the offspring is produced by two parents with the fusion of gametes. So, asexual reproduction is of 6 types: fission, fragmentation, regeneration, budding, vegetative propagation, and spore formation. And in sexual reproduction, we will study sexual reproduction in flowering plants and sexual reproduction in human beings. So, I have divided this chapter into 3 parts. In this first lecture, we shall discuss the asexual mode of reproduction. In the second lecture, we shall discuss the sexual reproduction in flowering plants. While in the third lecture, we shall discuss the sexual reproduction in human beings. So let's begin. So let's start with the different modes of asexual reproduction. First, fission. Fission is the splitting of organisms into 2 or more daughter cells. It usually takes place in unicellular organisms. Fission is of 2 types: binary fission and multiple fission. Binary fission is the division of the parent cell into 2 daughter cells. Examples. First, amoeba. So, in amoeba, the division of cells takes place in any plane. But in few other organisms, binary fission occurs in a definite orientation in relation with the structure. And example of such organism is Leishmania, and it has a whip-like structure at the end of its cell. And binary fission in Leishmania occurs in a definite orientation in relation with this whip-like structure. And Leishmania is nothing but the microbe causing Kala Azar. Second, multiple fission. So multiple fission is the division of the parent cell into many daughter cells. So in binary fission, 2 daughter cells are formed, while in multiple fission, more than 2 daughter cells are formed. That means many daughter cells are formed. Example of multiple fission is Plasmodium, which is the malaria causing parasite. Second, fragmentation. So it is the breaking up of an organism into 2 or more pieces. It takes place in simple multicellular organisms like Spirogyra. So basically, fragmentation is same as fission, but fission takes place in unicellular organisms, while fragmentation takes place in multicellular organisms. And let's see how Spirogyra reproduces by fragmentation. So Spirogyra breaks up into smaller pieces upon maturation. These pieces or fragments grow into new individuals. Now, such cell-by-cell division is not possible in complex multicellular organisms. So, such cell-by-cell division is only possible in the simple organisms and not in the complex multicellular organisms. This is because complex organisms are not a random collection of cells. In complex organisms, the cells are highly organized into tissues, and these tissues are further organized into organs, and organs are further organized into organ systems. Thus, these multicellular organisms need many complex ways of reproduction. We shall study about these complex ways of reproduction in the complex multicellular organisms in the third lecture. Now let's return to our modes of asexual reproduction. And the third mode is regeneration. So many fully differentiated multicellular organisms have the ability to give rise to new individuals from their body parts. Examples are hydra and planaria. So if an individual is cut or broken into many pieces, then each of these pieces can grow into separate new individuals. However, regeneration is not the same as reproduction. Now this is because these organisms depend upon being cut in order to reproduce. But however, if they are not cut, then they will not be able to reproduce. Thus, regeneration is not the same as reproduction. Fourth, budding. So as the name suggests, it is the process of reproduction through buds. And example is hydra. So in hydra, a bud develops on the parent organism. After maturing, the bud detaches from the parent body and becomes a new individual. So in this way, hydra reproduces by budding. And the fifth mode is vegetative propagation. So as the name suggests, it is the production of new plants from the non-reproductive parts like roots, stem and leaves. Advantages of vegetative propagation over sexual reproduction. So these plants bear fruits and flowers earlier than those produced by sexual reproduction. It makes it possible for the plants that have lost their ability to produce seeds to reproduce. The plants produced through this vegetative propagation have all the same characteristics as the parent plant, as they are produced by a single parent. While in sexual reproduction, there are many variations. Examples of vegetative propagation. First, bryophyllum. So in bryophyllum, the buds produced along the leaf margins fall on the soil and develop into new plants. So in this way, bryophyllum is able to reproduce through its leaves. Another important example is potato. So in potato, new plants arise from the buds present on the underground stem. Now, apart from these natural methods of vegetative propagation, there are some artificial methods of vegetative propagation. These are cutting, grafting, layering and tissue culture. Let's study about each one of them in detail. First, cutting. So a small piece of stem, root or leaf is planted again to give rise to a new plant. For example, in rose. So we just take a small piece of stem and then plant it again into the ground and it grows into a new plant. This is known as cutting. Second, grafting. So in grafting, we obtain a superior quality plant by taking two different plants and we take the root system of one plant and the shoot system of another plant and then tie both of them. So basically, if we want the characteristics of two plants to be there in a new offspring, then what we will do is that we will take the shoot system of one plant and the root system of the other plant and then tie the both. And in this way, we will get a superior quality plant. And example of grafting is mango. Third, layering. So in layering, the roots are artificially induced on the stem and then detached from the parent plant. For example, layering is done in jasmine plant. Another artificial method of vegetative propagation is tissue culture. So in this, a small part of tissue is cut from a plant and grown in artificial medium. Now, in the artificial medium, the cells divide rapidly to form a small group of cells called callus. This callus is transferred to a separate medium containing hormones where plantlets are produced. Now, these plantlets are sown in the soil to give rise to mature plants. So in this way, the plantlets can be artificially produced and then sown in the soil to give rise to the plants. And this is nothing but tissue culture. And the sixth mode of asexual reproduction is pore formation. So the thread-like structure that develops on bread if exposed to moisture for few days are the hyphae of bread mold. So if you leave bread for few days in the open, then you may have seen that some black spots develop on it. And these are the bread mold. Now, there are 10 tiny blobs on the hyphae called sporangia, which contain spores. These spores can develop into new rhizopus individuals. Rhizopus is nothing but bread mold. So in this way, bread mold reproduces by spore formation. This was all for today. In the next lecture, we shall discuss sexual reproduction. Thank you.